Duchenne muscular dystrophy, or DMD, is a progressive and disabling genetic neuromuscular disease which affects approximately 1 in every 3,500 to 5,000 newborn boys. DMD is a universally fatal disease which is characterized by muscle weakness, leading to delayed walking, abnormal gait, and frequent falls, among other developmental delays in the first few years of life. Over time, children gradually lose the ability to walk and typically succumb to respiratory or cardiac failure during their mid-20s. Within muscle, the protein dystrophin performs a critical structural role, anchoring the actin skeleton to the sarcolemma membrane in conjunction with other proteins of the dystroglycan complex. In doing so, dystrophin is thought to help protect the muscle from strain-related damage during muscle contraction. DMD can be caused by a variety of mutations to the dystrophin gene, which is found on the X chromosome. Such mutations can lead to a lack of dystrophin, which results in muscle damage, degeneration, and loss. The dystrophin gene is one of the longest human genes, spanning approximately 2.5 million bases. It is comprised of 79 different protein-coding segments, termed exons, which are interspersed amongst introns. To generate the dystrophin protein, the cell must first transcribe the entire length of the dystrophin gene into pre-mRNA. Within the cell, specialized cellular machinery known as the spliceosome binds specific sequences on the pre-mRNA and removes the introns to produce the mature mRNA. The dystrophin mRNA is translated by the ribosome using a reading frame that reads the mRNA 3 nucleotides at a time. Each 3-nucleotide sequence is called a codon, and each codon is translated into 1 amino acid. Amino acids are attached to the growing protein. Incorporation of the correct sequence of amino acids is essential to creating a properly folded functional protein. Most commonly, DMD is caused by frame-shift mutations due to deletions of 1 or more exons from the dystrophin gene. In these cases, the exons following the deletion are misaligned or out of frame, preventing translation of a functional dystrophin protein. Such frame-shift mutations result in production of little or no functional dystrophin protein. Lack of dystrophin leads to a cycle of muscle cell degeneration, inflammation, and fibrosis that is characterized by loss of muscle mass and muscle wasting.